So oxidative stress is an imbalance between the free radicals and antioxidants in the body. Under normal conditions, the cells are able to balance the production of the oxidants and antioxidants, and then oxidative stress occurs when cells are subjected to excess levels of ROS or as a result of antioxidant depletion. Now, it is harmful because ROS attack biological molecules such as lipids, proteins, and DNA that are involved in the pathogenesis of lifestyle related diseases. Then, oxidative damage to reactive oxygen species can be through membranes, DNA, and proteins. Like membrane is like lipid peroxidation, damage to membranes and lipoproteins, DNA. You have the DNA strand breaks, mutations leading to cancer. And for proteins, there's majorly aggregation and fragmentation, enzyme inhibition leading to oxidative stress, disease, and aging. Now, oxidative damage to proteins, it results in site-specific amino acid modifications, fragmentation of the peptide chain, aggregation of cross-linked reaction products, altered electrical charge, then increased susceptibility to proteolysis. Then sulfur-containing amino acids and thiol groups are very susceptible sites. The oxidation of iron sulfur by sulfur oxide destroys the enzymatic function. So it's the oxidative damage to proteins can lead to destruction of loss of structure function of essential proteins and enzymes, and the entire cellular metabolism is blocked. Now, antioxidants is a substance that delays or prevents or removes the oxidative damage to target molecules. You have all heard about the antioxidants. Like there are certain you apply on skin and something like that. So it prevents the aging and stress to the skin. So it is a very important because it does not allow oxidation to happen. So there is no universal best antioxidant. It's just the relative importance that depends on which, where, how the ROS is generated and what target of damage is measured. But oxidants or reactive oxygen species play a very important role in multiple diseases and can lead to different types of cancers. So it's important that antioxidants are important to neutralize or mop up the free radicals. Now, antioxidative defense involves the enzymes that are catalytically removes the ROS, superoxide dismutase, catalase, glutathione peroxidase, and peroxidases. You have certain proteins that removes the pro-oxidants like transferrin, ferritin, albumin, haptoglobin, ceruloplasmin. And then there are low molecular weight substances that are synthesized in vivo or you are obtaining from the diet in the form of vitamins and plant phenols. Antioxidants can decrease mutagenesis and thus carcinogenic in two ways by in decreasing the oxidative DNA damage and by decreasing the cell division. Now, proteolytic enzymes are enzymes that are involved in degradation of proteins called the proteases or the proteolytic enzymes. Now, industrially, these are much available and are produced by microorganisms that are usually the mixtures of endopeptidases and exopeptidases. So, endopeptidases are the ones that convert proteins into polypeptides, and exopeptidases convert the polypeptides into amino acids. That's the smaller fragment. Then there are a list of few proteolytic enzymes like papain, ficain, pepsin, rennin, trypsin. Now, peptidases, like I mentioned, are of two types: endopeptidases and exopeptidases. And these endopeptidases digest the internal peptide bonds, and exopeptidases digest the terminal peptide bonds to release the amino acids, like the aminopeptidase and the carboxypeptidases. So, proteins to polypeptides is basically endopeptidase, and exopeptidase is polypeptide to protein amino acid sequence. So there is a substrate that is present, so there are enzymes that acts upon it and it breaks the uh, amino acid or the protein into a C-terminal, N-terminal fragment. It breaks at certain positions, like the trypsin cleaves at the carboxyl side of arginine and lysine residues, whereas thrombin cleaves arginine glycine bonds in particular sequences. only. Therefore, these are certain proteolytic enzymes and they function very differently. Now, chymotrypsin cleaves on the carboxyl side of bulky aromatic and hydrophobic amino acid residues. Then you have elastase that cleaves on the carboxyl side of residues with small uncharged side chains. Protease enzymes functions are also very important and are essential in many biological processes because they are involved in the regulation of various metabolic and cellular processes. They are proteolytic, they help in digestion and catabolism of proteins. They catalyze the hydrolysis of peptide bonds and convert them to amino acids, which is then absorbed and utilized by the cells. They are required by the blood coagulation processes. Protease enzymes are involved in cell division, growth, apoptosis, and migration. Protein recycling and transport across membranes. Then they are involved in the activation of precursor proteins and zymogens. Proteases provide immune support and regulate the process of tumor growth, metastasis, inflammation, etc. And they help in wound healing and muscle soreness. So this is not what I will be basically going through. I will be directly going into viruses because last class we studied about. So this is mainly about a few motifs and all, but we have studied about the IDPs, so we need not worry about all of this. So if we talk about virus, so these are infectious particles. Then there is a genetic material and protein code surrounding it. So a virus is basically a small infectious obligate intracellular parasite, and these are not living. So viruses are chemicals and by themselves cannot reproduce, and a susceptible and permissive cellular host is needed for the reproduction of viruses. Now, viruses must make an mRNA that can be translated by the host ribosomes. So, if you look into the basic virus structure, it can be DNA or an RNA, and there is a capsid protein, so you get a nucleocapsid. Now, this nucleocapsid is basically a naked capsid virus. Now, this naked capsid virus is surrounded by the glycoproteins and lipid membrane. So, the spike proteins, like for a coronavirus, and that. So, then you have an enveloped virus inside which you have the capsid, nucleocapsid, and then surrounding you have the glycoproteins surrounded by a lipid membrane. So the capsid symmetry can be like helical or icosahedral. So these are the shapes of by which the capsid looks. Then there is an attachment that happens. So attachment can be through tropism. There is a lock and key model. Then it allows it to sit and then it penetrates. Then disassembly, where the viral genome is secreted in the cells, it transcribes to an mRNA and leading to the formation of viral proteins. At the same time, the viral genome also multiplies, leading to multiple number of viruses or the capsid proteins, and then there is basically release. So for a retrovirus, how it happens is that there is genetic material inside the virus. There is entrance, then uncoating. So the single-stranded RNA inside is uncoated. You have a reverse transcription because it's a reverse transcription using the reverse transcriptase. You get the double-stranded DNA and it travels to the nucleus. It integrates with the host DNA and then the provirus to the transcription of the viral protein. So the outcome of the cellular infection is somewhat like this. You have a virus, there is an attachment to the cell, then it penetrates. It can just remain like that and lead to transformation into tumor cells, tumor cell division, and transformation of the normal cells to the tumor cells. The other way out is that it starts multiplying by the previously maintained process. The cell dies and via lytic infection, and but the virus has already grown and it has survived. Therefore, it just basically moves out. The third is by the slow release of virus without any kinds of cell death. So it's a persistent infection. So you are undergoing through an infection. There is a viral count that's increased, but the cell does not die. So it's not harming the cell in man. And the other one is the latent infection, where the virus present but not causing any harm to the cell later emerges in a light infection. So the immune response is like a cell-mediated immunity that is important for the recovery from viral illness, or antibodies that are important for the protection from repeated exposure. Like the next time when the virus attacks, it should not be able to cause any kind of effect. Then it's important that you understand about the number of variants versus the time it takes. Okay, so number of variants is the acute infection that happens in days. Then there is a persistent infection that remains for over months or years, and then there is a latent infection that is sudden rise. Okay, so yeah, that's about the thing. Now we can talk a little bit about the motives over here. So like I was telling that there are, so we had till now we always wanted to discuss about the structural proteins, the structured proteins, and things like that. But a very important protein that plays a very important role inside the cells to regulate the different cellular mechanisms and all are IDPs. In molecular biology, IDPs are intrinsically disordered proteins. It lacks a three-dimensional structure or three-dimensional motifs. Now, the three-dimensional structure is typically absent of its macromolecular interaction partners such as proteins or RNAs. So IDPs are things that interact with nucleic acids as well. So they are fully unstructured to partially structured and includes random coils like molten globule-like aggregates or flexible linkers in large multi-domain proteins. They are sometimes considered as a separate class of proteins along with the globular, fibrous, and membrane proteins. Now, IDPs are very large and functionally important class of proteins, and their discovery has disproved the idea that 3D structures of proteins must be fixed to accomplish their biological functions. For example, IDPs have been identified to participate in weak multivalent interactions that are highly cooperative and dynamic, lending them importance in DNA regulation and cellular signaling. So we can tell that the distinctive function structures sequence interactions are always not very important for controlling the function. Even a disordered protein is equally eligible to go for carrying out a function. So yeah. So from the sequences, so these are the some of the notations and all. Don't want to go into all of these depth because these are much more complicated things. So how can you predict? So like if we think about the disorder database, there is Disprot, you have MobiDB. This will give you an idea about the prediction of the sequence. You give the sequence and it can predict it. Nowadays you have like so many predictive tools that will help you to understand whether a protein is structured or not. Then from the by the sequence you can understand like whether the protein will fold or not and something like that. But nowadays these predictive tools have made things easier. You don't need to manually predict it. You can just do it. You can put the sequence and it will do the rest of the work. And as biological roles of these disordered proteins, you have the allosteric regulation, enzyme catalysis, then Many disordered proteins have binding affinities with their receptors regulated by the post-translational modifications. Then the intrinsic disorder is particularly enriched in proteins implicated in cell signaling and transcription, as well as chromatin remodeling functions. And genes that have been recently been born de novo tend to have higher disorder. Then there are some flexible linkers that want to be attached to the proteins, and these are very very important for the allosteric regulation, then auto inhibition procedures, and also that controls a lot of the cellular mechanism and cellular signaling. Then as linear motifs, these can be like slims, so short linear motifs. So these are also disordered sequences. And the most difficult thing about that is that unlike the globular proteins, they do not have active pockets. So they bind using a particular sequence, or folding upon binding. They fold upon binding, and the structural characterization of the linear motifs are very very difficult. So these IDPs are very difficult to predict the structure because they are very transient, flexible, and that is why doing an NMR for a structural study of these proteins are quite complicated. But apart from that, the functions of these proteins are very much so. Yeah, it's quite interesting to know about or study a lot about these proteins. But over here, we do not need to know all of that or anything about that. So, yeah, whatever we had like about the proteins, we were able to complete in last week's class. So in our today is the last week lecture. Tomorrow we will have a special class or a special extra session of 2 hours where we will like do a few doubt clearing and all. Like I guess you people have received a mail from NPTEL about the sessions. So from 6 o'clock I have it. So there I will be talking a little bit about like proteins, and so that is also part of the lecture because uh, one class we talked about the recombinant DNA technology. So yeah, tomorrow let's see, we will touch about protein. We studied about the protein interactions. We will talk about protein, carbohydrates, lipids. So there also we will come across IDPs because we have will have protein nucleic acid interactions. But we do not need to know about in details for this particular course. So I'm not going into the depth of all of that because then it gets a lot more complicated. So what you need to know is basically like about the peptidases, the antioxidants, the peptidases, the proteolytic enzymes, where they cleave. These are very very important portions, like trypsin, where it cleaves chymotrypsin and all. These are very important, how it cleaves and some of the functions, and then a little bit about IDPs, and then we go into the viruses, about the mechanism of how viruses attack and how it can multiply, undergo transcription, translation, replication, assembling, and all, and then finally release. And the one of the finest viruses we have known now about is the retrovirus, and how we can stop them, or what are the ways of that. Okay. Okay. So any questions to here? Okay. Maybe I will give 5 minutes to see if you guys have any questions. So, should we go with the question and answers? So we can start with the question and answers then. Fine. So the first question says, which one of the following oxidized forms of cysteine is most likely to be directly linked to protein damage? Sulfonic acid form, sulfone form, sulfonic acid form, or sulfenic acid form? Which one is correct? Any answer? No? Okay. So the sulfenic, or option D, is the most likely one to be directly linked to protein damage. So because it is highly reactive intermediate during the oxidation of the thiol group, that's the cysteine residues, and it plays a key role in the redox signaling and oxidative stress. That is why sulfonic acid form is the problematic one. Okay. Dityrosine and nitrotyrosine are produced from tyrosine in the presence of various reactive oxygen species. The two biomarkers are generally detected by dash and dash spectroscopy. So, which of the two spectroscopy can be used to detect uh, dityrosine and nitrotyrosine? So if you see that the dityrosine is, so if you look, like dityrosine has a very characteristic absorption band in the UV visible spectrum, due to which you can detect it in the UV visible spectroscopy, whereas nitrotyrosine is a fluorescent molecule, and its presence can be easily detected using fluorescence spectroscopy. The next one says, which of the following reagents is not used for analyzing N-terminal of peptide sequence? Which of the reagents is not used for analyzing N-terminal of peptide sequence? So what are the names of phenyl isothiocyanate? That is, it's Edman's reagent. 1-fluoro-2,4-dinitrogen benzene, that is Sanger's reagent, and dancyl chloride is all. So all of them are used for N-terminal amino acid analysis of peptides. But cyanogen bromide is used to cleave a peptide chain, that is specifically at the C-terminal side of a methionine residue. That is why it's not used for N-terminal analysis. The next one says, which of the following statements is true about Ebola virus? So let's see what we know about Ebola. So we know that we want know a few things like Ebola virus is a single-stranded RNA virus, and it's not a DNA one, and it is just a minute. So the Ebola virus is a single-stranded RNA virus and is also not circular in nature. The virus genome, it encodes 7 proteins, including the matrix proteins that are important for the budding process, and it helps the virus to adopt the different shapes. So if you look over here, that the first statement is incorrect, and also option 4, that is statement 4, is also incorrect because it's not circular in nature. So option 2 and 3 are correct, that is option C. So this is much easy, like where you have to, this is a virus, icosahedral virus, and you have to label A, B. So what is A and what is B? So most of you are aware of the coronavirus and all. We had suffered a lot in during that stage. So A is basically the, so what is that? So this is an icosahedral virus. So A is fiber and B is hexon protein. Which of the following processes do not or does not occur in HIV? Replication, reverse transcription, translation, transcription. So you can see that reverse transcription and transcription both does not occur in HIV. Okay, maybe not. Wait. Sorry. So transcription and translation. So 1 and 4. So replication and transcription does not occur in HIV. Reverse transcription and translation, reverse transcription and translation happens in HIV. So 1 and 4 is the one. So replication and transcription does not occur in HIV, and you will find reverse transcription and translation to be seen in the HIV life cycle. Okay, so there was a bit of confusion regarding this. Anyways, so unknown peptide protease cleaves the peptide sequence. So there is a peptide sequence given into three polypeptides. So the name of the specific protease. So you see that it has cleaved after phenylalanine, after tyrosine, after phenylalanine, and after tyrosine. So which is the protease that cleaves in these specific sites? So there is a protease that cleaves after the aromatic residues, so that is chymotrypsin. Okay. The next one it says, so the polypeptide is there that's cleaved by thrombin and trypsin. What are the digested fragments? So you have a thrombin and trypsin. So what will be the digested fragments? So trypsin cleaves after lysine. So we will get GLK and PMRWA. So that is after trypsin cleavage. And thrombin cleaves after R. So we get PMR and WA. So you will get three fragments. One is GLK, PMR, and WA. The next one says the tryptic digest of an octapeptide consisting of two lysine, two methionine, one serine, one proline, and one glycine, and one arginine. So it yielded two tripeptides and one dipeptide. So both the tripeptides do not contain arginine. Which of the sequence is correct? So it means that one of the dipeptide has arginine. So so you have to just work out this thing. So you have lysine. So you know where does trypsin cleave. So it means that the tripeptides do not contain arginine. So if you look into option B, so trypsin cleaving after lysine and arginine, you would get KPK, MSK, and GR. So that over here you get two tripeptides and one dipeptide, where the dipeptide does not have dipeptide has the arginine, but the tripeptides does not. For option C, you will find that it cleaves after R and K, so K and R. So you will get like two tetrapeptides. Okay, for option C. So it will cleave over here. So you will get a tetrapeptide. And for the other cases it will cleave over this portion, so it is a hexapeptide, and also over here this is an hexapeptide. So only option B is correct. Which of the following amino acids are preferably present in intrinsically disordered proteins and play a role in preventing the formation of a stable structure? A very direct question. I was discussing about the types of amino acids that can be there. So it's glutamic acid, proline, and arginine. Which among the following is not true about intrinsically disordered proteins? So IDPs are proteins which lacks a stable three-dimensional structure. That is correct. IDPs are disordered only in localized regions. That is also possible. So it's correct. IDPs carry low densities of charged amino acid residues. This is incorrect because it contains so many amino acid charged residues that because of which it is actually disordered. And IDPs exist as highly flexible conformations. That is also correct. Now, tau protein is polymerized into paired helical filaments and straight filaments, forming neurofibrillary tangles when the protein is highly what glycated, phosphorylated, acetylated, or hydroxylated. So tau proteins become hyperphosphorylated, which leads to aggregation into paired helical filaments and straight filaments, forming neurofibrillary tangles. This is a key process and is a pathological hallmark in Alzheimer's disease and tauopathies. So phosphorylated is the correct one. And the next one says which amino acids are prone to carbonyl derivatives due to oxidative stress. So which group of amino acids? So oxidative stress, we see that it can lead to formation of carbonyl derivatives on different residues like tyrosine can get hydroxylated, methionine gets oxidized, cysteines form disulfide bonds or other oxidized forms. Therefore, tyrosine, methionine, and cysteine will be the correct group. Which is incorrect about the fly casting mechanism of IDPs? So, what does fly casting mechanism of IDPs indicate? So the fly casting mechanism is a model for how the IDPs bind to their targets. Unlike the rigid proteins, they search for a specific binding site at first and then binds non-specifically and folds into a final high affinity conformation. The non-specific binding reduces the search space for the final specific binding, which is a characteristic of this mechanism. Therefore, option A, that is specific binding sites searched for and found while folding, is the incorrect one. And the last one is which of the following compounds cannot act as an antioxidant? So what are antioxidants? The antioxidants are molecules that can prevent or slow down cell damage by neutralizing the free radicals. So they typically have phenolic OH groups or OH group attached to benzene, or there are other oxidizable groups that can donate the hydrogen atom. If you look into the first compound, it has a catechol group, that is two hydroxyl groups on the adjacent carbons, which is a well-known antioxidant motif. If we look into option 4, this is a phenolic hydroxyl group, which allows it to function as an antioxidant. Option 3 will also be true because there are so many hydroxyl groups that are attached. Option 2 cannot be correct because it's an amino acid cysteine with a thiol group. While thiols act as antioxidants, the compound should not be able to function properly and it does not act as a nickel antioxidant. Okay, so option only, so B, that is only two will be the one. Okay, yeah. So I'm done. Thank you. If you have any questions, you can go ahead. And another time, I'm just mentioning like tomorrow there will be a special class or an extra session on like there can be doubt clearing, or if you have any questions, special questions, you can ask me, and I might go through a few important parts that I feel you guys should know. So yes, then okay. If you have any questions, you can go ahead. No questions? Okay. Thank you very much. We will again meet tomorrow for the final session.